as I had said last time, is sort of the classic view of that's basically the only way to make transcription, which is true. So we also use compaction and positioning, and including positioning, binding to the matrix and those kinds of things. But of course, to accept that requires a paradigm shift from the classic. So we had used as our example the promoter of INS, and how we have a number of different promoter elements that are going to be involved with the regulation of INS expression. And especially what, of course, we drew our attention to was the KLF one there, KLF11 binding site. And that's a specific sequence then of nucleotides that binds a particular transcription factor, KLF. And we talked about how transcription factors are going to bind onto the outside of these particular sequences of nucleotides. And that allows us to form a deoxyribonucleoprotein complex. And we'll see that it's more complicated yet. And that's then involved with the regulation of transcription in ways that we don't totally understand yet. So the classic view, of course, is the train on the track model, as we had said. And for the train on the track model, the idea would be that in some way you have RNA polymerase IIs floating around haphazardly throughout the nucleus. And when they happen to bump onto a promoter that has the right assembly of transcription factors, then you bind RNA polymerase II and set it up and get it ready and let it go. And it comes on down the track transcribing that particular gene. And if I say that in a sarcastic way, it's because it can't possibly work, yet it's taught as dogma. So it's like, okay, but it can't work, which is ridiculous. So, ridiculousness aside, how does it actually work then? Well, apparently we have transcription factors, of course, that are found within our chromosomal domains, and we have the loop chromatin alter those transcription factors. There's a picture which showed that illustrates this using looping based on the binding to the matrix. But whether you look at the matrix or not, we still have to loop the particular chromatin regions onto genes and set them up and get them ready to go. And so transcription factors are going to be involved with that in some way, and we don't know exactly how yet because people are going through just under a false paradigm. So in some way, though, we probably are going to actually hook up the region that needs to be transcribed to the RNA polymerase II containing the RNA polymerase II of the RNA polymerase II containing transcription factory, and set it up, and then let transcription proceed, and probably in a regulated fashion. All the evidence indicates it's a regulated fashion. And then we are going to sort of spool the DNA through the RNA polymerase II, and probably all regulated as well. And we know, for example, the promoter stays associated, so there's probably even regulation there. And of course, all these things haven't been discovered, haven't really been delineated yet. But as I showed you back when I was showing you pictures of looping of chromatin and so on, the evidence is very clear. The promoter stays associated with the RNA polymerase II, which would be yet another problem for the train on the track model. Because if that was the case, then you'd be sort of like, how would you be motoring at that point? You'd be motoring, but you'd be pulling the promoter with you as you motored along the chromatin, which would not be a very likely process, given the crowded nuclear environment, which is again be kind of ridiculous. But in some way, probably the part at least of the old model holds. That is, we have to set up these whole systems such that we have the appropriate start site identified, and we have bound the chromatin up appropriately at the start site to the RNA polymerase II. So that probably is working. Now, there's probably other things going on. We don't have any knowledge of what is the transcription factor, what are its components. It's highly likely that within the transcription factor, we have particular regions that are going to be involved with binding to transcription factors and setting that up and so on. But until somebody looks at it, we won't know. But highly likely that kind of thing will be happening too. So that much at least is sort of similar to what we had talked about before, and similar to the classic model. Now also, and this is some of the more interesting thing that sort of allows us to illustrate some of our concepts that we've been talking about. Also, we have a situation where the transcription factors are probably involved also with the attachment to the matrix. So of course we define the matrix attachment region binding proteins, and so there are numerous matrix attachment regions along the chromatin, some of which are in genes, some are out of genes, and those kinds of things involving how we bind up the DNA to the chromatin, which of course to transcription of the genes found in those areas. And transcription factors are probably involved with that too. And again, it just sort of, we need more evidence. So could it be that transcription factors are in part, at least some part of the whole system of binding to the matrix? Probably, because they're involved with regulating transcription. So in some way they might interact with matrix attachment region binding proteins, or we may find out that some of them are matrix attachment region binding proteins. We classified them all as transcription factors just because they were found to bind to promoters, but that doesn't mean that they're not also involved with binding directly to the matrix or indirectly, perhaps by interacting with matrix attachment region binding proteins. So probably all sorts of things like that, and again, remain to be delineated. So we'll see. We just need to have people asking the questions and doing the research and so on to figure out if that's going to be the case. So the matrix is probably involved. We just need to figure out how and so on, how that's regulated. Another thing that's interesting, I think, with respect to the concepts that we just talked about, is that we're discovering that we make a whole complex at the promoter, and so it's not just transcription factors. So if you were to consider, say, the way the book talks about it, it's like you bind a series of transcription factors, set up the right polymerase II, and let it go. That's really the classic model. But it's not just binding transcription factors. We bind transcription factors, and those transcription factors facilitate the binding of other transcription factors, or maybe inhibit the binding of some. So you form like a complex complex. I like that phrase. So it's elaborate, and so probably what we should do is to sort of adjust our thinking to think along the lines of the promoter as a region at which we are going to assemble a complex, a dynamic complex. So very similar to, say, the way we had talked about something like the spliceosome. You have things coming out and things interacting and things moving around. It's a dynamic complex that's being formed. And so it's not just transcription factors sort of linearly arrayed, although there's some aspect of that. Obviously, linearly along the chromatin we have particular transcription binding sites, but we're going to bind a whole complex there. So to give us sort of some of that flavor, I drew this picture for you, and it has some additional information on it too. And this is what they actually look like, what promoter regions would actually look like, of course, just depicted in a cartoon sense. So here we have a gene, so this, you could imagine this is something like, say, INS, because we use INS as an example. And I've only shown here a portion of the promoter. It would actually be longer thinking about all those elements and so on, but just to give you an idea. So we have some transcription factors bound in different areas. So each one of these Easter eggs that I drew here, and didn't think of them as Easter eggs until this week. These peeps that I've drawn there with, I wish I had a color picture of this. I wish I could Easter eggs, change it for different holidays. So we have transcription factors bound to the chromatin. So notice here, transcription factor bound there. So you can sort of imagine to yourself then that for this particular Easter egg, it's going to bind to it with your sequence nucleotides there, and this one binds to that sequence there, and this one there, and this one there, and so on. So we're binding transcription factors, which essentially are defined as those proteins that do bind directly to things like promoter elements. And then we also bind additional proteins onto those. And so here I'm showing some additional proteins that are bound onto those. So like here's an additional protein, here's one, here's one, here's one. And they're not exactly transcription factors because they aren't binding directly to the promoter elements. They're binding to proteins that are bound to the promoter elements. So these things are sort of like part of the complex that's being formed at this promoter to help regulate the process of transcription. We can call those transcriptional coactivators. That's what they're being called now. So the idea is that they're going to be involved with the regulation of gene expression. Typically, the way people talk about transcription factors at this point is that they think about them in terms of their ability to sort of turn on a particular gene. However, some of them might turn it off. So, I guess if you wanted to try to delineate that, you would say, well, some of them are transcriptional co-inhibitors or something like that, and that's cool. But that kind of thing has sort of got us bogged down in some other concepts as well that we'll be talking about dealing with cancers. So, we don't know what all they do, and they probably do complex things. But one thing that's definitely happening is that we're using a complex of various proteins there, including transcription factors bound to promoter elements, but then also proteins bound up to those transcription factors and bound to each other, forming this complex, which appears to be quite dynamic. And so there are other things bound here as well that are involved in the process of regulation.